to 2005, there were about 9,000 papers released in the leading journals, but still the knowledge here in India is in the rudimentary stage. Now only we are making it, we will see. Bacterium like Bifidobacterium, Lactobacillus, Clostridia certain specimens, group comprising of the gut microbiota complex, they are digesting non-digestible carbohydrates found in the dietary fibers, producing short-chain fatty acids. These short-chain fatty acids going to get the expression of G protein coupled receptors 41, 43 and 10A on L cells of the intestine, which will be emanating GLP, endogenous GLP-1 and P-peptide, PWI, and decrease the PPR gamma activity, which enhances oxidative metabolism in the liver and adipose tissue, decrease the fat accumulation, mitigating the fat accumulation, thereby the lipotoxicity is reduced. When the lipotoxicity is reduced, there is an absence of cetramide, the triacylglycerol and diacylglycerol, which will clear a good liver function, though the insulin sensitivity will be more. Insulin resistance is not there. So this GLP-1 polypeptide WI decreases appetite and prolongs the gastric emptying, increases the peristalsis, and insulin sensitivity increase, insulin secretion increased, beta cell apoptosis prevented, and there is a lot of beta cell regeneration and good amount of beta cell function. So butyrate promotes the expression of tight junction. You know, butyrate is also one of the short-chain fatty acid. It promotes the expression of tight junction proteins, occludin, zona occludins and singulins, etc. And they will produce more expression of mucus secreting goblet cells. So thick mucus is produced, and they will close the gate of the junctions, which is guarded by these proteins, and prevents leaky gut syndrome. So butyrate, not only that, because of certain action on the epithelial integrity, prevents the entry exit of LPS, which we lipopolysaccharide, which is an endotoxin, will be alluding to, which will be cause for the emanation of cytokines and chemokines and lipokines, especially IL-6, tumor necrosis factor alpha, thereby producing inflammatory milieu and increased insulin resistance, which is prevented by this short-chain fatty acids, butyrate. When short-chain fatty acids meet type 2 diabetes, how many minutes or more? 1.5 minutes. So, short-chain fatty acids not only produces the expression of the protein coupled receptors on the L cell, producing more GLP-1 and WI, they will be producing more amount of beta cell function and prevents the formation of LPS. You know, whenever there is a gram-negative bacteria is more in the intestine, you could understand the wall of the gram-negative bacteria secretes LPS, lipopolysaccharide, which is an endotoxin. This LPS, when it expressed on the toll-like receptors 2 and 4, which are transmembrane receptors, they kindle the endocannabinoid system 1 and 2, traverse through the junction, and when the endotoxin traverses through the junction and enters the circulation, there is a condition known as metabolic endotoxemia. This metabolic endotoxemia is a root cause for pro-inflammatory state, which will be producing abundant amount of insulin resistance, thereby producing this thing, diabetes mellitus. These are the thing. Secondary thing, the common salts are protection against conversion of bile acids to secondary bile acid, and prevention of the disposal of the emanation of branched-chain amino acids, aromatic amino acids. So these two processes will be prevented by common salts, which are stimulated by the production of the branched-chain amino acids, aromatic amino acids, which are caused for intestinal So, dysbiosis, decreased butyric secreting organisms, increased permeability, changes in the bile acid pool, decrease in the bile acid catabolism, increase in the production of branch-side amino acids, increased chain of amino acids, which will be producing insulin resistance. So, absence of gut microbiota, which are very good for internal milieu, they will be producing insulin resistance, leaky gut syndrome, and the decreased immunomodulation, thereby producing exorbitant insulin resistance and production of the diabetic milieu. This can be mitigated by the prudent use of what we call it as prebiotic and probiotics and good dietary fibers. The prebiotics are the food to the probiotics. Probiotics are living organism which will substitute the deficient organism in the gut. So, naturally occurring, 1 minute. Naturally occurring prebiotic and probiotic, you have to know: tomatoes, artichokes, onions, garlic, chicory, dandelion greens, asparagus, leeks, berries, bananas, flaxseed, legumes, and pitta, white or red of this dragon fruit. They are all very good prebiotics and probiotics. Probiotics, when you come here in the naturally occurring probiotics, sauerkraut, kimchi, kombucha, yogurt, cottage cheese, and miso soup, and all the fermented bread, which we call it as sauerkraut, and this bread, all will help for preservation of probiotics. So when the prebiotics and probiotics join together, we call it as symbiotic. So prebiotics, probiotics, symbiotics are heavily available nowadays for usage along with the regular treatment of diabetes mellitus. Microbiota metabolites like short-chain fatty acids, strong influence in diabetes mellitus. Though there are various relations with the host metabolism impact on several pathways. There is a bidirectional relation whether the diabetes mellitus impairs gut diversity or robustness, or the gut dysbiosis produces the diabetic milieu.
So I
there
think
are a
we
lot
can
of
conclude the
ongoing
session,
research is going on. There's going to be a couple of international conferences, one in Venice, another one is in Japan, and in October, November. And in our own country, AAG Hospital of Hyderabad, under the head of Dr. Nageswar Reddy, is doing a lot of research on the gut microbiota, which will be paving way for future therapeutic arsenal.
Thank you, sir.
Very much.
Thank you.